course, I didn't know that I was dreaming at the time that I had this experience.
What is the meaning of dreams?
I have no idea.
It is delirium. It is not like delirium, it is delirium.
Waarom dromen we? En waarom zijn onze dromen vaak zo bizar en ongeremd? Schuilt er een diepere, maar verhulde betekenis in onze dromen? Waarvan zijn onze dromen eigenlijk gemaakt?
Dreaming is a peculiar psychological experience that occurs in sleep when the brain reactivates. The brain first deactivates at the onset of sleep, and then it reactivates periodically throughout sleep at about 90-minute intervals. In fact, the brain is always activated partially, so dreaming can occur in any stage of sleep. But when this dynamic and dramatic activation process that we call REM occurs, then dreaming is likely to be intense and prolonged, detailed, and have all the characteristics that we associate with.
Onze slaap verloopt in afwisselende fasen van diepe slaap en remslaap. In dit stadium van rapid eye movement sleep kun je aan de snelle oogbewegingen van een slapend iemand zien dat hij op dat moment waarschijnlijk droomt.
How can we do research on dreaming?
Well, it's relatively straightforward. We wake subjects up and we ask them what was going on in their mind before they were waked up. We collect the reports. Now, we can do that in a sleep lab, or as we prefer now, we can do it at home.
Can we do it at home?
We can do it at home by using a nightcap, which we actually developed in this laboratory. What the nightcap does is to take signals that the EEG people think is noise and treat them as signal, in particular movement itself. And this is a technical detail, but the recording of eye movements, for example, is absurdly abstract in the sleep lab. They attach electrodes to the outside of the eyes and then measure the voltage difference between them. Well, that voltage difference is given by the retinae, and the retinae move toward one side, you get a negative potential, they move to the other side. This is a very, very expensive and indirect way of measuring movement. I mean, movement is visible. You can put the eyelid up and see the eye movements. And if you leave the eyelid closed, you can see the eyelid twitching. So in the nightcap, home-based sleep monitor, eyelid monitoring becomes the target of interest, and we get a lot of data.
Tot het PET-scan-onderzoek van Dr. Brown en Dr. Bolkin.
We conducted a series of studies using PET, positron emission tomography, to characterize the various stages of sleep and wakefulness. The reason we did that is because most of the information about sleep up until about 5-10 years ago came from EEG data, electroencephalographic information, which is very important in characterizing sleep stages, but the spatial resolution is very poor. You can't really tell where you are in the brain, and it looks like the whole cortex is responding monotonically in the same way throughout the various sleep stages. So what PET can tell you is relatively non-invasively what's happening inside the brain in specific regions, and the relative spatial resolution, the sharpness of the image for PET is much, much better than for EEG. We had to insert two catheters in, one a intravenous catheter, another an arterial catheter. The arterial catheter is for sampling blood during the PET scan so that we can actually quantify blood flow units. That's very important because the absolute changes in blood flow are very dramatic during transitions between sleep stages.
While they were sleeping and in the scanner, we would monitor, identify the sleep stages, and when a sleep stage of interest, stage REM, rapid eye movement or dreaming sleep. When they occurred, we would call for radio-labeled water. And Dr. Brown would go in and inject the radio-labeled water, and about 90 seconds afterward, activity would show up in the brain and we would begin collecting data for the scan itself.
O15 water is used because it traces blood flow. Blood flow is useful because it's coupled to neuronal activity. So the more an area of the brain is active, the more blood flows to that area to provide nutrients and to carry away waste. So the PET scanner acquires data from inside the brain. The areas that are more active retain more of the O15 water because blood flows more to those areas. So they emit positrons. Positron is a little piece of antimatter. And when it meets an electron, it annihilates and produces two light beams, two photons at 180 degrees apart. You collect all this information from all of the brain using the PET camera, and the camera then uses software to back project that information to a three-dimensional image of the brain, which is characterized by relatively more or less photons, the more or less the neurons in those areas are. And that the half-life of O15 water is about 2 minutes, so we acquire data for about 4 minutes in total. And hopefully the patient doesn't go into another sleep stage during that period of time, which often happen. What you're seeing is that at the onset of sleep, the transition from awake to slow wave sleep, deactivation of the pons and midbrain, the cerebellum, the basal ganglia, and the prefrontal cortex and inferior parietal cortex, also the cingulate cortex and the midline. Prefrontal cortex is involved in some of the highest order functions of the waking cognitive processes during wakefulness. It integrates information from all other regions of the brain and allows one to plan, to sequence actions in a goal-directed fashion, to inhibit certain actions that are socially inappropriate, to symbolize, to monitor oneself, and so on. These are the areas that are shut down. The brainstem, which drives activity in the cortex, is shut down as activity is decreased at the onset of sleep, but then becomes quite active during REM sleep. The limbic cortices, the perihippocampal and hippocampal gyri, and the cingulate cortex become wildly active during REM sleep. The transition from slow wave sleep to REM, you get activation of the pons here, the cerebellum, activation of just the midline portion of the prefrontal cortex, basal ganglia, the visual association areas, auditory association areas, and the frontal cortex and parietal cortex remain deactivated. And then once you wake up following sleep, this is after, compared to 20 minutes after waking, you get reactivation of the prefrontal cortex and reactivation of the parietal cortices. One interesting thing is that the inferior parietal lobule and the prefrontal cortex are involved in what's called working memory during wakefulness. And working memory is a type of immediate memory that allows you to retain things online from moment to moment, second to second actually, so that thoughts or emotions are provided, it provides a context or a through line for thought and emotion. Well, this process is interrupted during REM sleep because these areas are not active.
So you can't remember?
You can't remember from second to second, which can account for some of the bizarreness of dream imagery. Things morph into one another. If I dream of a cigar, it might turn into a locomotive. That is because I can't retain the previous image in working memory and provide a narrative through line for it.
What happens if people have a malfunction of the prefrontal cortex?
There's a famous case of Phineas Gage, who had a tamping iron blown through his ventromedial orbital cortex, preserving the rest of his brain, preserving his life. And he had a very characteristic set of symptoms in which his behavior was socially inappropriate. He had been polite before. He was now rude. He was disinhibited. He displayed one of my favorite neurological symptoms, which is Witzelsucht, which means bad jokes, telling bad jokes. But generally, so it's that ventromedial orbital area that's sort of involved, as being involved in social cognition, in mastering all these impulses and inhibiting those that are socially inappropriate. And there's some PET studies that have shown that this area of the brain is relatively less active in sociopaths or in people whose behavior characteristic is antisocial, characterized as antisocial.
Well, I think there are three phases to dream science in the modern era. One is the Freudian era, which was from 1900 to 1950, say, in which it was widely believed that dreams were the reaction of the mind to the activation of unwelcome impulses. I think that theory is dead now. And it gave way to the sleep science paradigm, the sleep laboratory paradigm, in the early 1950s, after the discovery of the reticular activating system and the capacity to record human subjects with EEG. So that period lasts from about 1950 to 1975, and it has to do with waking people up in sleep lab, getting the reports, trying to correlate the reports with measurable aspects of physiology. Perfectly reasonable. It just failed. All it showed was that there is a correlation with REM that is stronger than the correlation with non-REM. But it couldn't go very far. We needed the animal model. And the animal model started to be explored before 1975, but in 1975 we published our papers. So naturally, I mean, I think that's a watershed point. We showed that REM sleep was associated with and probably caused by changes in neuromodulators. The minute you have that model, you have a completely new way of thinking about dreaming. You say to yourself, dreaming occurs when the brain changes state. The brainstem manufactures molecules. We call them norepinephrine, serotonin, for example. And they're very important to the waking brain. They help us keep our thoughts in a row, to have recent memory, to have attention, to be able to think critically, to be able to think at all. Yeah. And without these, then the brain cannot do those things. And in REM sleep, those two specific chemicals are taken away altogether. That's a big surprise, and that's probably our most important scientific discovery, was the absence of the cells that make that stuff stop firing. So there is no release. Now at the same time, there is another chemical called acetylcholine, which is also important to waking, but it becomes reactivated in REM. So we have part of the chemistry reinstated, but not all of it. And we think that the difference between the states is largely attributable to that, including the regional changes of the brain. Because what these people with PET scans are measuring is blood flow. They're not measuring activation directly. They're measuring blood flow, and activation is inferred from that. Well, in the periphery of the body, what controls blood flow? Norepinephrine, serotonin, acetylcholine. So we think that the chemical model will prove to be the mother of models, but that remains to be seen.
And can you measure the amounts of chemicals in the brain in the various states of...
Only in animals, not in humans, because most people don't want you to put, cannulae into their brains.
In what way do dreams relate to psychosis, hallucinations?
Well, they're formally identical. I say not only similar, but identical. And boy, people have criticized me for that. But look, you have visual hallucinations. You see things perfectly clearly, but there's nothing there. It's all done inside the head. Naturally, you believe in these things because seeing is believing, right? I mean, and you believe a lot of other stuff that couldn't be true too, like I'm flying, you know? Because I have the sensory motor experience of flying. So as far as my mind is concerned, I am flying, right?
Right.
Well, but that's psychosis by definition. It's hallucination plus delusion. I've got it already. But let's go on down the checklist. I've also got orientational instability,
Yes.
the discontinuities and incongruities. I've got intensification of emotion, especially, interestingly, elation and anxiety. And I've got terrible memory. I can't remember what happened while I'm dreaming 3 scenes before. I wake up, I say, oh, yes, I have some recall, but there were 3 scenes before that I can't remember. You know, so this is not only psychosis, but it's delirium by definition. It is delirium. It is not like delirium, it is delirium. And you know, when you get what is called delirium, you get it by messing around with these chemicals. I've said that already, but it's so important to realize that. If you want to get delirious, either drink too much and try to stop, take amphetamines and try to stop, take LSD, you know, take ecstasy. All these things mess up the chemistry in such a way that REM is enhanced.
Why are you so lifelong intrigued by dreams?
Well, there are 2 answers to that question. One is personal. I mean, I just love them. I think they're wonderful. I mean, where else can you do what you want and have such mysterious kinds of experiences? It's not easy. And it's drug-free, and it's legal, and it's cheap, and all of that. But as a professional, it seemed to me that, you know, how are we going to study mental illness? It's very hard to do that directly. There's no good animal model for either schizophrenia or depression. But dreaming is a great model for psychosis. We've already talked about it. It has all the qualities of psychosis: hallucination, delusion, emotional intensification, blah, blah, on and on. So, if we can figure out how the brain, the normal brain, makes this normal psychosis, then we've got at least a bridge to something to stand on. As now all we need to know is, why do these guys dissociate? Why do they have dream experiences during waking? Well, an obvious answer would be they can't keep the neuromodulatory lid on the thing, you know? Why don't we dream during waking? Because the dream machine is suppressed actively by the neuromodulators. So, I mean, I'm not saying we understand schizophrenia, but depression is already very, very closely linked to sleep and dreams. You know that.
Go to sleep, my baby. Close your pretty eyes. Angels watching o'er you, peeping at you, darling, from the skies.
What is the meaning of dreams?
I have no idea. I don't know. I mean, the dream symbols can, I suppose, be meaningful in that they relate to, they can be used in a way that's meaningful, and that they relate to daily residua, you know. They may be, have something to do, the content may have something to do with memory consolidation in itself. I don't know.
The meaning is on the surface. It's transparent. It's there for the taking. You don't need to involve yourself in complex decoding to get use of dreams in psychotherapy. I mean, I do psychotherapy all the time. I talk to people about their dreams. It's straightforward.
What you see is what you get.
What you see is what you get. You get a lot.
100 jaar geleden publiceerde Sigmund Freud zijn befaamde Traumdeutung. Valt er voor zijn omstreden theorie nu nog wat te zeggen?
One of the things we haven't talked about is Freudian theory. It was thought that dreams probably are very important. I think Freud thought mainly to keep subjects asleep because they wanted to they were able to monitor unwelcome thoughts or images or emotions and then disguise them. The crux, I guess, of Freudian theory is that dreams represent some sort of wish fulfillment. And our data are not inconsistent with that. The co-activation of these limbic areas, which I talked about playing a role in memory consolidation, also would be areas involved in emotional processing. And the fact that that occurs, those areas are dramatically active during REM, was not inconsistent with that part of Freudian theory. But our data are absolutely inconsistent with the idea that there's a sensor or a monitor who is watching for the emergence of unacceptable wishes and disguising them in symbolic images. That, if any part of the brain would be responsible for those processes, it would be the prefrontal cortex, and the prefrontal cortex is asleep.
Freud used disguised censorship to account for bizarreness. We don't have to do that anymore. So we don't need disguised censorship. Should we use it? Why? I mean, what for? There's so much blatant meaning on the surface of dreams. Talk to anybody, you'll see it. Your brain is filled with chemicals. You can alter them, not entirely at will, but you can alter them enough to create all sorts of interesting experiences for yourself. Why go out on the streets and pay for some stuff you have no idea what the quality is and put it in your mouth and let it go into your brain? I mean, it's crazy. It's really nuts. You know, all these experiences are very interesting. I'm an ecstasist myself, but I get it without risk and without cost.
Maar liefst 3/4 van de bevruchtingen eindigt niet als baby. In de hoop de kans op zwangerschap te vergroten, is men in het Academisch Ziekenhuis Rotterdam op zoek naar de allerbeste embryo's. De bedoeling is om embryo's genetisch te keuren voor ze teruggeplaatst worden in de baarmoeder.